He has brought Noni to India, then it is come to the light, and we also introduced Noni by him. Then we have given a project to work on the availability of Noni and also other Morinda species. So therefore, we got a first project by Health India Laboratories, then to survey the Morinda species in South India. So we have went different places in Tamil Nadu, Kerala, Karnataka, and we could see the presence of about 6 different species of Morinda. In addition, Morinda citrifolia in the western coast of Kerala up to Mangalore. Then by the time we were not aware of the disease problem, and because generally we believe when the plants are in the wild natural stand, we don't look at very carefully. So therefore, not much information were brought in India about the pest and diseases occurring in Noni. And moreover, the Noni, as you see here, it is very hardy crop, grown in different condition. It grows in acidic soil and alkaline soil, drought condition, even including saline soil, even in the sea water. So therefore, generally it believes that it's a hardy crop, therefore it is free of diseases. You can see here, it is also growing in rock, and it's also extensively grown in a forest location. This is in Hawaii. And it has various abiotic resistance. But once if it is introduced, any crop is introduced as a monocrop, as a domesticated plant, then there will be the disease problem because of the various environmental factors as well as the availability of the pathogen probicules. So slowly the pathogen may establish on the crop and try to infect it. So therefore, when we started screening the presence of Noni in different parts of the South India, we would like to also see the occurrence of diseases. This is for our curiosity, because as a pathologist, I instructed my students, Dr. Surendran as well as Dr. Sagadevan, to find out the occurrence of any diseases in the wild stand initially. And we put a question, is Noni has any disease problem under natural habitat? Or by then, that time, the Noni Cultivation Council has started. They wanted to introduce Noni in India. So therefore, whether if it is introduced in a cultivated field, by the time the Salavakam NRCN has started, they have introduced Noni plants from Mangalore, Calicut, and also some accession from Andaman has come, they have put in. So then we wanted to know whether any disease start occurring in this the natural stand as well as the cultivated plantations. Then we found to know, yes, Noni, though it is considered as a hardy crop, it also susceptible for various pathogen and nematode. Then we will look at to the literature. Initially, we didn't have any information about the disease occurrence in Noni. So therefore, we went to the literature. This is the information we got it from literature. There are foliar diseases like black flag, anthracnose, which is also considered as a leaf spot and leaf blight, then stem blight, then stem canker, sooty mold, short hold, and algal leaf spot. These are the foliar pathogens. Foliar diseases have been recorded in elsewhere, not in our country. Then root diseases like root knot, nematode, associated wilt, and also post-house disease, that is rhizopus fruit rot. There has been information, we got it from the literature, mainly through Dr. Scott Nelson. This is the very important disease, has been identified as a problematic in Hawaii and Fijian island, has been identified by Scott Nelson, caused by PHYTOPHTHORA BOTRYOSA. This is another important disease called anthracnose, caused by Coleotrichum species. You can see the extensive damage of leaves. Then yet another disease called a stem blight, caused by Sclerotium ralsei. It also started infecting the root and move on to the stem. Then this stem canker, even so far the pathogen has not been identified. It's also extensively damaging the crop in Hawaii Island. Then sooty mold. Then start hold. There is initial leaf spot as occurring, and then the leaf has been damaged and the hole has formed. This is a short hold. Then very interestingly, algal leaf spot caused by Cephalosporus is very interesting. The fungal algal pathogen, it's also causing the leaf spot in Hawaii. Then root knot, it's a very major issue, both in Hawaii as well as in Fiji and some other Pacific island country. Sometime back we also had this problem in our country. Then again, this is the rhizopus fruit rot. So these are the information we got it from the literature, mainly through the publications of Scott Nelson. Then by the time we also wanted to know whether any the disease occurrence in our country. So therefore we made a survey with the support of the World Noni Research Foundation. It's from 2004 to 2006. We made survey in Kerala coast. Then we found out there is a leaf spot, leaf blight caused by Fusarium species. We also able to isolate and identify the pathogen. Then another leaf spot called Colletotrichum leaf spot. Then sooty mold, but it's not very extensive. Again, the short hold. Then there is a viral symptom also. Very interesting, we could also see this kind of fruit rot could be bacterial or fungal pathogen. There is a soft rot in nature as well as the dry rot in nature in the natural wild stand. It's very interesting. By the time we were not aware of this disease, later only we came to know the dry rot and soft rot as occurring in a big way. But this was actually we recorded in the natural stand between 2004 and 2006. Then root knot nematode. Then we also made survey recently in Andaman with the support of WNRF, and Dr. C.R. Singh from Andaman has helped us in going around the different parts of Andaman and also the natural stand in plantations. And we could see the occurrence of leaf spot. And Dr. C.R. Singh was mentioning that there is not much problem, disease problem, but yeah, that's true, not much disease problem except the leaf spot. It was occurring in patches in severe form. And also this kind of fruit damage, again we could see. But unfortunately, we could not isolate any disease, pathogen from this leaf spot. It may be a physiological disorder or maybe a scorching effect. So then we also made a survey in Pacific Island countries. Thanks to WNRF, they sent us, myself and Dr. Siddhananda went different island countries. We could also record, it's our record, not from the Dr. Nelson. We recorded the occurrence of PHYTOPHTHORA BOTRYOSA flag black in Fiji and Hawaii, which was in severe form. Then anthracnose disease, again caused by Colletotrichum species, Fiji, Hawaii, and Tahiti, which occur in moderate form. And leaf spot and short hold, Fiji, Hawaii as a moderate form, and Tahiti is rarely occurring. Then dieback, it was first time we could record this occurrence of dieback. It's identified, but the local scientists whom we met in South Pacific Secretariat, they were telling that it's a viral disease, but we were not established the pathogenicity because we could not bring the sample, but it is in Fiji with very, very severe form. It's nothing to do with the flag black symptom. Then very interestingly, the stem gall also we could record in Fiji because of maybe viral infection. Then this is again unidentified black fruit. There is a fungal growth on the fruit which is in a severe form. Almost 40 to 50% of the fruits in the tree have been affected by this symptom. Then again there is another parasitic problem. Even this Casita filiformis or Cascuta, these are the two parasitic plants also occurring in the Noni tree. In Morinda tinctoria also, it is a common occurrence in India. And the left side photo has been taken from the Scott Nelson website, and right photo we took from Hawaii. And disorder, coming to the disorder, there are different disorders apart from the diseases we could see. That is that nutrient deficiency. Then it is a form of chlorosis, we can easily find out, and stunted growth will be there. Because of that, the yield has been reduced. And very interestingly, if high level phosphorus is there, this kind of uneven ripening is occurring. But we could not see much in the plantations. But this is occurring. Actually, we have taken this photo in Hawaii, and sometimes in India also. I have seen one of two places we could see uneven ripening. That might be the excess phosphorus condition. Then this is kind of a fruit cracking, very commonly occur in Fiji, Hawaii, and Tahiti Island, and sometimes even Noni farm also I have seen maybe some physiological disorders. Very interestingly, Noni plant, though it is considered as a very hardy plant to grow in a different condition, it is very, very sensitive to cold. So it is a purely tropical plant. It is not occurring in subtropical or temperate region, purely tropical. If the temperature goes below 12 degrees centigrade, this kind of injury is occurring, the plant will die. And we could also see in some location in North India, you can see this plantation has been severely affected by the cold injury. Previously, maybe a few days before, that temperature was about 4 degrees or 5 degrees, so therefore this has been damaged. And interestingly, even if you above ground is damaged, the below ground part, it is retaining, and then temperatures shoot up, then again it is sprouting. That is very interesting to know. So you can see severe form, this cold injury. So we have to take care before planting the temperature parameter. So you can see closer view, the entire plant has been died. Then survey of Noni plantation, we made some survey of late, then we could see the occurrence of the soft rot as well as the dry fruit rot, which are very, very, very severe form. Among these two fruit rot, the dry fruit rot occurs in severe form, almost more than 40% fruits have been damaged. So therefore we could not get marketable fruit yield because of this problem. So therefore it is become an alarming issue in recent past. And so therefore we need to take care of this fruit rot disease. And then the later, so WNRF also started funding various projects, even for crop protection. So they have funded 3 projects so far. One is to Dr. Kannayan from Sannagro. The another project is to Nakiran, Marimuthu and Raghuchandar from TNAU. The third project to me, so to study various aspects of the disease problem in India. And Dr. Kannayan has surveyed in entire South India the occurrence of Noni diseases, especially the wild stand as well as the plantations. So he went around Karnataka, Andhra Pradesh and Tamil Nadu, and he has listed different Noni diseases like leaf spot, short hole, leaf blight, zonate spot, target spot, and also ring spot and southern blight. Then algal parasite. Interestingly, he also recorded algal parasite and wilt, and then rhizobus fruit rot. And very interestingly, he also recorded occurrence of fruit rot in India, in addition to few other diseases. So these are the diseases. Actually, he has surveyed and listed about occurrence of 8 different foliar diseases and 5 different stem or root diseases, and the occurrence of root knot nematode, then 2 different fruit rot diseases, and there are other physiological problems. So then he has made a survey, and based on the survey, he has given a major diseases in South India like leaf spot, short hole, leaf blight, zonate spot, and target spot. These are the important diseases according to his survey. And Kannayan also made effort to find out a viable control measure, and he made different combination of biocontrol agent like biocontrol biocontrol agent, and also in combination with the organic manure he tried, and he found out the treatment 6 comprising of organic manure and different combination of microbial product is very effective in controlling the Noni diseases. So he has recommended to go for this treatment for application. The next project carried out by the Tamil Nadu Agriculture University by Dr. Nakiran et al. So they have concentrated again survey in different parts of South India, mainly Tamil Nadu, Karnataka, and to some extent Andhra Pradesh. So extensive data has been collected, very voluminous data, and they found out these 2 pathogens like Alternaria alternata causing leaf blight as well as the Coleotrichum cleosporidis causing anthracnose. This is a major issue, that's what they figure out. And very interestingly, they also found out these 2 pathogens also responsible causing the fungal dry fruit rot. Then in addition to soft rot caused by a bacterial pathogen, Pandoia agglomerans. So then all the pathogen they systematically done and identified through molecular characterization. Then they tried the organic way of controlling the plant diseases, Noni diseases, using Pseudomonas fluorescens, Bacillus subtilis, and Trichoderma viridi. They did in vitro as well as in vivo the screening procedure. Then also taken this treatment, best treatment to the field, and they found out application of these bioagent will certainly reduce the plant disease. In addition, they also found out the bacillus species, identified the different bacillus species, which are very, very effective in controlling the Noni pathogen alternaria as well as the other pathogen. So then find out the mechanism. They also elucidated what are the ways these biocontrol agents act against the pathogen, and also they optimize the growth of biocontrol agent, grow in a large quantity, and developed formulations. And these formulations they tested in the field condition. And very interestingly, the foliar spray of bacillus isolates could reduce significantly the Noni diseases in the field condition. That was the result carried out by Nakarian et al. Then another project given to us. Under this project, we also made survey in different locations, 5 different locations, including Andaman we made, and we found out systematically the occurrence of the soft as well as the dry fruit rot, which are now become a major issue. And we could see the occurrence is very high during November and September and August, mainly in the Calicut accession, not in the Mangalore. But soft rot occurrence was not very, very significant compared to dry fruit rot. Therefore, we wanted to identify the pathogen responsible for the dry fruit rot, and we could isolate a pure form. And we also established the pathogenicity using the coach postulate under the moist chamber, which we developed, small experiment. And in plant also we have tested within 9 days. If we inoculate the pathogen, it's able to completely degrade the fruit. So the fruit has become rotten. So therefore, without any doubt, we proved that the pathogenicity of the fungus, whatever we isolated from the. Then we went further to see the histopathology. Then we could also see the pathogen penetrate inside and also causing the necrotic cell death, and the mycelium also we could see inside. Then we wanted to find out, we also seen the morphology, and finally we went to identify through molecular characterization using ITS primers, and we sequenced the ITS gene sequence, amplified genes, and the data we have submitted to NCBI with the number KP340801. It is available already. Then we also constructed the phylogenetic tree. Finally, we identified this as a Neocytalidium dimetiatum. This is the pathogen which is responsible for the fruit rot, dry fruit rot, occurring in large extent. And this is also known as a fruit rot pathogen in different crops. So then we wanted to find out the control measures. Then we wanted to see any bacterial microbial biocontrol agent can be used for the control of these diseases, the fruit rot, soft rot, and we could isolate different rhizobacteria, around 160 rhizobacteria we could isolate from the soil sample collected from Noni farm, and also about 32 pilosphere bacteria we isolated from the leaf as well as the fruit sample through washing techniques. Then we screened systematically against the dry root rot pathogen. Then we found out 33 isolates showing activity out of 106. Out of 33, 9 were able to show high activities. Therefore, we selected these 9 for further study to find out the mechanism of action. Then we could see 7 isolates could produce hydrophore in way of competition, and 2 isolates produce protease and chitinase, and 4 isolates could produce beta-1,3 glucanase. But unfortunately, none of our 9 isolates able to produce hydrogen cyanide. Then these bacteria mostly are rhizobacteria, so therefore we wanted to see any growth promotion effect, because if it is there, probably we can use as a dual purpose, improving plant growth as well as inducing systemic resistance as well as the plant growth promotion. And out of 9, 5 isolates were able to produce EPS, so that this EPS can help in maintaining the soil moisture. All the 9 isolates, very interestingly, could produce IAA. So therefore, they are able to improve the plant growth. They have plant growth promoting trait. Out of 9, 6 were able to produce ammonia. Then we could see out of 9, 1 isolate they were able to solubilize the phosphate, and 3 were able to solubilize zinc, and 4 were able to solubilize the potash. So therefore, based on the biocontrol traits as well as the plant growth promotion trait, we were shortlisted to 2 organisms, two bacteria, rhizobacteria. Then we wanted to identify in addition to a bilosphere bacteria based on the activity. So identification done based on the 6' RNA sequencing. We were able to identify Bacillus cereus, Bacillus amyloilicipation, both are rhizobacteria, and 1 bilosphere bacteria, interestingly, they are Bacillus amyloilications. We made formulation and permeation also we tested in plant growth promotion, and the combination of Bacillus cereus, Bacillus amyloilicipation able to promote the plant growth in the seedling condition. Then field experiment also recently we conducted, just the nutshell. the application of foliar spray of Phyllosphere bacterium, that is Bacillus amelolicupation, could reduce the root rot disease incident very significantly and also improve the marketable yield of noni fruit. So in summary, noni is susceptible for various pathogens. Leaf blight, leaf spot, and nematode associated wilt were common occurrence in different parts of the world, including India. Dry fruit rot has emerged as a major disease, and its pathogen were identified. Microbial biocontrol agents have been identified, formulated, and evaluated in the field experiment. Therefore, noni diseases can be managed using biocontrol agent. Coming to the very interesting part, the World Noni Research Foundation also brought out a very wonderful the publications in the form of technical bulletin as well as the abstract book. One is by Kannayan, that is the technical bulletin, deals with the various survey of noni diseases in India, South India. And then Siddhananda et al., we made a very wonderful handbook. Actually, that handbook is considered as a very important for identification, field identification of plant diseases. Based on the symptom, you can identify what are the pathogen, what are the diseases. So it's another interesting book. And Ratnam et al., myself, and also Dr. D. R. Singh, we made a book and we collected all the information until 2012, whatever happening in noni research. We compiled all the abstract, and in which crop protection also one of the chapter in which we have mentioned a lot of information about the noni diseases. So we can get information from there. And Nakirian et al. Also published another technical bulletin. It's a very extensive technical bulletin. Will give a lot of information about this. Thank profusely for the organizer for giving me the opportunity to present my paper. Thank you very much.